Well, I've been working in cancer research for 20 years now. I started doing a PhD over at UCL on breast cancer research, and at the time that we started that research, a lot less was known about the biology of breast cancer, and we've continued with the research that we started, looking really for markers of aggressive breast cancer. As the research has developed, we've adopted new techniques. We've really started to study the cells as well as the lots of tissue samples and specimens from patients. So we've gone from the basic biology right through to the sort of, I suppose, what people would say translational research. The motivation, of course, is that whilst treatments for the primary breast tumour are much more advanced now, there's still a huge problem with the development of metastases. This is the development of secondary cancers. We would really want to try to find new ways of targeting both the primary tumour and the metastatic deposits to try to prevent the mortality that we have from breast cancer and other solid tumours. The main problem following cancer, particularly breast and other solid tumours, is this secondary spread of the cancer to distant organs. On a personal level, the motivation, well, I think everybody's been touched by cancer. I've had family members, you know, two of my aunties have had one pass away from breast cancer, one has breast cancer, a cousin has breast cancer, so I have a very personal connection with breast cancer. But I think most people now have an understanding of the problems of cancer, and they can relate to the drawn out and quite often very unpleasant side effects of the treatments, and that they can understand the need to push forward the boundaries. So. We are looking at proteins in the blood. We're doing some work looking at the tumour tissue itself, and then we're doing work looking at blood samples, or actually fractions of the blood, which we call particular fractions of the blood, in this case serum. We're looking at the proteins because the cancer cells produce unusual proteins. They produce increased amounts of certain proteins, and they also there's also modifications on those proteins or changes on those proteins. And we're particularly interested in the changes on the proteins, and the focus of my lab is the changes in the glycosylation, or the sugars on those proteins. This is quite a complicated area. It's taken a while to get the method so we can really work on this and get things sensitive enough to be able to test in the blood, where you have very low amounts of certain proteins from the cancer in the blood. If you can measure this in the blood, what we're looking at in my lab is whether the measurement of certain proteins and their sugars on those proteins, whether that allows us to tell earlier whether the cancer has spread to other parts of the body. And that way we understand that would give an opportunity to treat the metastasis earlier. And this is really what we're wanting to do. If you have to wait for imaging of the tumour in the bone or the lungs or wherever, the argument is, of course, it's quite advanced by then. Whereas if you have a blood test, a very sensitive, specific test, then you can intervene earlier. And of course, that's to the benefit of the patient. So we're very lucky because we hold a wonderful, wonderful collection of samples from about 3,000 women who have breast cancer in the UK. When we have their blood, we have their urine samples as well. And ultimately, we would like to look in the urine so that we could make a urine test, which would be less invasive. At the moment, we're working on the blood test. It's looking very promising at the moment. Well, in the case of breast cancer, the first thing normally would be to use surgery to remove the cancerous mass. if the cancerous mass is too large, often there will be a chemotherapy first to try to shrink the tumour and then surgery. My surgical colleagues also remove the lymph nodes under the armpit to work out whether the cancer has metastasized at the time of diagnosis. And there's been great advances in looking at the lymph nodes under the armpit. As well as looking at removing it surgically, the patient then is normally will be offered post-operative radiotherapy to the breast. Once that's been completed, they would often, depending on the status of their tumour, be offered some sort of hormonal therapy. And in quite a lot of patients, they will be offered chemotherapy. That's normally after the surgery, they'll be offered chemotherapy. Now, the most recent treatment is this biological treatment, which is the trastuzumab, the antibody treatment, or Herceptin treatment. Very, very expensive treatment, but we know that it's targeted to the patients who have the increased level of that receptor, and that approximately 50% of those will respond to the treatment. So this is incredibly promising for us as scientists, because we're trying to find our new molecular markers, the proteins, and we want to personalize the medicine so that patients are not unnecessarily given medicines which can have huge side effects, of course, and affect quality of life and such. The work is important work, because if you can identify the cells which are showing changes in protein levels or enzyme levels, then you can look at making very, hopefully targeted and specific therapies, whether they're inhibitors or whether they're other types of biologics that could go in there and prevent that enzyme function. Then you've got the potential to get a new class of anti-cancer compounds. And of course what we want to get the compounds that are going to target the cells which are really important for the development and continued regrowth of the tumour. So we want to find the cells which are the aggressive ones, which is of course what you're focusing on. So your project is important. I think it will have some very, very interesting outcomes, and I think it's also proof of principle, which is an important, very, very important thing there. If you find something in one type of cancer, some changes, as we know, say, from the protein kinase work, that these changes are then common across a range of cancers. And we know this also from the biologics that have been developed. So what's true for one type of cancer will often be applicable to other types of cancer, which is what's so important here. Point. I think most people can appreciate how personally how awful it is sometimes to be faced with a diagnosis, and then well, hopefully you have the hope that the treatments will work, but if you've gone through a number of rounds of different treatments and then you find that there is not anything that can be done after that, then people are vulnerable to claims, and you know, sometimes there's a sense of wanting something but it's not necessarily there, and they will be wanting to go for some treatment sometimes which perhaps haven't been proven. So I think it's really important that with any of these new developments that we don't oversell them, that we are measured in what we're saying. You know, we are saying that there's some really interesting science, there's incredible developments that we've seen and that we're continuing to see, and of course that's something for the future, and that there will be clinical trials, and that hopefully there will be properly randomized controlled clinical trials, and that people will eventually benefit from that. But there's a real problem with overselling and giving hope where it may be 10, 15, 20 years before some of these initiatives are properly realized. Some of them are coming sooner, but I think that that's the important thing sometimes that is lost in the debate about some of these new technologies and say, oh well, this is a new wonder cure, this is going to cure everyone for their diseases. And of course it takes a long time to get a product from initiation right through to market, and there are a lot of there's a lot of attrition, there's a lot of casualties on the way as you go through that whole process of, we know this from our experiences with pharmaceutical industries, know this only too well. So I think it's wonderful that we have the potential. I think it's really important. Obviously I'm running a lab and we're really excited about the work we're doing, but we also want people to realise that we're not rushing into this, we're taking it steady, and you know that is the important thing. We don't want nasty mistakes. We want to take it steady so that people can really ultimately benefit from the research.